Okay, so question is, do you need to know the different polymerases? The answer, no. So A, that's factual information you could look up during this exam if you had time. So you don't need to memorize that. And also, we didn't cover that in class. Thank you, though. That's a great question about the exam. Yes?
I just have a question about the exam. Is it set up exactly the same way it was last time?
Okay, format of the exam. Is it set up exactly the same as it was last time? Very similar. So the idea, there was a technical issue last time, which is why everybody had to access it through Blackboard. It was supposed to go out through Google Classroom. It didn't. This time it should, hopefully, all go through Google Classroom. You get the PDF there, you annotate it, you attach it to an assignment back to me. No emailing, hopefully. And I've changed a few other minor things about the exam, mostly timing. So, since you asked, the individual portion is going to start the exam. It's going to be 25 minutes, like last time. There will be 10 questions for that 25 minutes. They're all essentially multiple choice, fill in the blank, label, give a brief one-sentence description of something, interpret data, do a calculation. So it's not just factual recall multiple choice, but they're all fast to answer questions. There's no extended writing or anything like that. So after that's 25 minutes, there will still be a 5-minute transition before the group work. And then the group exam is going to be 15 minutes. This is where the biggest change happened. It's not very big. I wrote 3 questions. The group has to answer 2. So you get to choose, which may be more difficult. I don't know. I'll be interested in your feedback afterwards. It means there has to be some discussion in your group about which questions to answer. But hopefully that will save you some time and let you answer questions that your group feels more comfortable answering. It also means that you don't have to answer a question on every topic we studied in preparation for this test. So I asked 3 questions, your group answers 2. Other questions?
So I just want to be clear, when you're crossing for 2 different traits, you said it was standard practice to assume wild type. Like, remember you were saying, like, if you had like a roller worm versus a glowing worm, you assume the wild type for the glowing part of the roller worm.
Exactly.
And wild type is always homozygous dominant?
Wild type, so if I give you a question that has something to do with, I'm talking about 2 different traits. Let's say I'm talking about individuals that have Drosophila, red bristles, let's say red hair, and white eyes. So if I tell you that you've got a red, and both of these are the mutant versions. So red bristles, let's say, is mutant, and white eye is mutant. If I tell you you have a white-eyed line of flies, you assume that you have the wild type bristle color in the same strain. That's a good assumption to make. If I say white eye and I say nothing else about the fly, you're going to assume it's wild type at the bristle gene. And you could come up with whatever allele designation. Usually we use a plus to indicate wild type. So you could say it was plus over plus diploid at the bristle gene. Now, you can't make an assumption about dominance. So whether or not the red allele at the bristle gene or the wild type allele is dominant, we have no information. If you felt like you had to make an assumption, you could, but that's a dangerous assumption to make. We have to do a specific type of experiment to determine dominance. So what would we do in this case? If you wanted to know, so let's say the plus allele is, what color are Drosophila bristles normally? Black? Let's just say it's black. So plus allele gives you black bristles, and R gives you red bristles. How are we going to tell which one's dominant? A monohybrid cross. So what does that look like? Okay, so a monohybrid would be homozygous. Your monohybrid cross, you'd start with a homozygous parent, plus over plus, and you'd cross it to homozygous for the other. So what are the genotypes of the F1 generation? Genotypes. Every single one of those offspring is going to be something slash something. Plus slash R. They inherit a plus from one parent, they have to. That parent only has plus versions of the chromosomes. The other parent only has Rs. So all the F1s are plus over R. So the plus over plus P0 had black bristles. The R over R had red. Okay. Somebody say one of the two following words. One person. Say one of the following two words. Red or black. Just one of you. Say it loud. Black. I heard black first. So let's say the F1s are black. All of the F1 individuals, every single one, has black bristles. Which allele is dominant? Plus. Because when you're heterozygous, when you've got one version of each, you want to know what's the phenotype. Whichever phenotype that F1 individual has reveals which allele is dominant. So you never know. The wild type allele is not always dominant, though. It's a common alternative conception. We'll see some examples in class later where the wild type allele is not dominant. Other questions? Yeah.
Can you show us the difference in the separation when you have the bivalent chromosomes for meiosis 1 and meiosis 2?
Okay, so how the chromosomes separate, chromatids separate at meiosis 1 and meiosis 2.
Yeah.
Okay. What ploidy of organism should we do? Choose an easy one. Diploid? Okay, let's do diploid. I mean, we could do tetraploid, but we've only seen diploid up to this point, and I don't want to add any more confusion if I don't have to. So we'll redo diploid. Okay. So if each of these lines represents a double-stranded piece of DNA, a chromosome, what does this look like after synthesis? It's temporarily tetraploid, which means there are 4 copies of this chromosome. So here I'll draw a little key. So that line, it's double-stranded DNA, we're going to call this chromosome 1, just for fun. Okay, so now we've got 4 copies of chromosome 1. One was inherited from the mother, one was inherited from the dad.
After synthesis, do they specifically, or do they specifically look like that, or have that X shape? Are they still covalent or bonded in the middle?
So after synthesis, at some point, the chromosomes do reassociate with each other, so that you do have, sometimes they're drawn like this. the two newly synthesized chromosomes are associated with each other physically, so that you know that the two versions that came from mom are next to each other, and the two versions that came from dad are next to each other.
Would it be wrong to draw the both mom chromosomes on X shape and the dad things next?
It doesn't matter if they're in a line or crossing each other at this point. Yeah. You can draw them however you want. Except what happens next. Here's the part where maybe you can't draw them however you want. So if the cell is going to do the first meiotic division, division 1, this is meiosis. How do the cells line up across that dashed line, which is where the cell's going to divide?
So on one side you'll have the dad chromatids, and on the other side you'll have the mom.
So I heard on one side we've got the dad chromatids, and on the other side we have the mom chromatids. Does anyone disagree? Sir?
When they separate, it always goes to mom and dad and dad. Can it be mom and dad and mom and dad?
At this stage in meiosis, it always segregates like this. So the two copies of the same allele, mom and mom, always go into the same product after the first division. Product meaning daughter cell. And the two versions of dad's chromosomes always go into the other one. I'm going to say something very controversial now. You won't sound controversial because it's only controversial to geneticists, but they can hear me if they ever watch this video. Ultimately, to you, probably, it doesn't matter. Because at the end of meiosis, regardless of how the chromosomes segregate now, if you put two chromosomes, any two chromosomes in one daughter cell at this point, and any two in the other, what's going to happen after the second division? You wind up producing 4 haploid gametes. It's the exact same outcome. But that's not how it works in biology. So to a biologist and to a geneticist, it matters that this be done the way it's actually done in the cell. 2 copies of mom go into one side, 2 copies from dad go into the other daughter cell. But as far as we're concerned right now, that doesn't actually affect the outcome of meiosis. There are some subtle ways it does, but so this is what happens after the first division. That's
question
interesting.
one, basically just asking metaphase 1. So.
Okay, so this is which question?
Question 1, figure below shows the nucleus of diploid cell before S.
Oh, on the study guide?
Study guide.
Yeah.
Yeah, so you're just asking for.
Right. So this is how these chromosomes line up in meiosis at the first division. What would this look like? Well, here, we'll do second division. So you've got 2 cells. One's got 2 copies of mom's chromosome, and one's got 2 copies of dad's. Now there's only one way to arrange the chromosomes in the next products of division. Each chromosome gets each cell gets a single chromosome. So haploid gametes. Where on this screen would you refer to a double-stranded piece of DNA as a chromatid and not a chromosome? At what point in time is it that the chromosomes become chromatids, and when do they go back to being chromosomes again? Remember, this is totally arbitrary. It's just nomenclature. Right, so after synthesis, so what was a chromosome, after synthesis is called a sister chromatid. And then at what point, so then there's sister chromatids for a while, and at what point do they become legitimate to a geneticist chromosomes again? After the second division. So it might be interesting to note, or might not be, you tell me if it's interesting to note, that it's every time I've drawn the two sister chromatids with a dot in between them, showing that the two versions of the chromosome that have the same allele are next to each other, where they're called sister chromatids, because they're sistered together, they're paired up together. When they're not physically associated like that is when geneticists refer to them as distinct chromosomes.
So they're called like Tetrads, so that means one, like the two, the sister chromatids would be four
Right.
in each.
So where do we have the tetrad? That's the division right before the division of meiosis 1, where all four of the sister chromatids, the two from mom and the two from dad, are all lined up together in a group of four.
But they are being pulled to each side.
Before they're separated after the first division. Yep. So at the second division, what do we call that blue pair of sister chromatids? It's no longer a tetrad, it's a dyad. And this book introduces a term, I think it was this book, introduces a term I had never actually heard before, which is what do you call a single sister chromatid? A monad. You can make up whatever names you want, I guess. So, other questions? Yeah?
So is there a difference between a dyad and what is it?
Bivalent. Yeah, a bivalent, bivalent. Same thing. Dyad equals bivalent or bivalent. Yeah. That's a group of two, the two sister chromatids that are from the same parent. So for example, new color. In yellow, the pair of sister chromatids that are both dad's alleles would be called a bivalent or a dyad. Yeah.
Can you go through the division of an embryo? Because it's a study guide. You talked about how many times are they going to replicate and how much of the original DNA is where?
Okay, so question about one of the questions on the study guide. Let's see. That was question 5. You start with a single cell embryo. It divides 3 times, or undergoes 3 rounds of division. How many strands of DNA are the original strands, or what percentage, rather? Okay. So yes, this wasn't, embryonic development is not something that's part of this class, but I included it because it's simple, it's cell division. So if you have one cell that goes through 3 rounds of division, how many cells do you wind up with? So 1 becomes 2, 2 becomes 4. So each of those divides. 2 becomes 4. Each of those 4 divides. You have 8. So like any cell division process, embryogenesis is the same. Do cell division, if you go through 3 divisions, you get 2 to the 3, or 8 cells. How many strands of DNA do we, let's assume this is a 2n equals 1. It's diploid, but it's only got 1 chromosome. How many chromosomes do I draw in this just fertilized 1-cell embryo? How many double-stranded molecules? 1 chromosome. 2. So it's got 2 double helices. 2 versions of chromosome 1. One came from dad, one came from mom. How many original molecules of DNA, how many original strands of DNA? That's 4 strands. Hopefully everybody's with me so far. 2 strands per double helix. That's why it's called a double helix. So if we zoom to the end, how many strands of original DNA are going to be remaining in after that 3rd set of cell divisions? Careful, trick question. We start with 4 strands that are original. How many original strands are there after 3 cell divisions? 4. They're original. So the real question is 4 original strands. Still. So to figure out something like what fraction of the total number of strands of DNA are original, we just have to figure out how many strands have been synthesized in that period of time. Okay. So what are the total number of strands if you have 8 cells that are diploid? How many strands are in each cell? That hasn't changed. It's still the same as it was in the original cell. 4 strands per diploid cell in this case. So we've got 4 strands, 4 strands, 4 strands, 4 strands. So there's a total of 32 strands. 4 of them are original. Why did I ask this question? To test if you can do fractions? No. To make the point that you never get rid of the original strands of DNA. It's part of it's a DNA replication question. The more cells you have, the lower the ratio of original strands to newly synthesized strands since life began, but you never get rid of those original strands of DNA. Other questions about this? Sure.
Each of the 4 original strands are going to be in different cells, right?
Right. So you wouldn't be able to necessarily, you could tell me which 4 cells, but that would just be a guess. And I wouldn't know for sure. Related question. So the question, in case you didn't hear it, was how are those 4 original strands distributed among these cells? So if I had colored them red, for example, where would you draw the 4 red strands after that 3rd cell division? Would there be 1 strand in 4 different cells? Could it be possible that all 4 of the original strands wind up in 1 cell? Or is there a particular way they would have to be distributed after the 3rd round of cell division? So how does DNA replication work? Semi-conservative. Step 1, you denature the 2 strands. So each of those things becomes single-stranded. Then you synthesize the complementary strand, which is base paired. And then what's going to happen after synthesis? You go through the cell cycle and you go to mitosis. What happens? cell division. So you necessarily put the two original copies into two different daughter cells. So there's no way to get a cell that has more than one original red, in this case, strand of DNA in it. So you could decide which of these 8 cells to put an original strand of DNA in, but you can't put more than one in one cell. And I guess we're all lucky because since we're not testing on probability, I couldn't ask you a question like, what's the probability of getting, anyway. We won't worry about that. Yeah.
Wasn't there a way where there's mutation, where one cell doesn't have any strands and the other one has two strands much? You're talking about something like that. So then you can have 2 original strands.
Excellent point. So what's a situation in class where we talked about something that could go wrong, where you might have more than one, we don't expect it. If the process of DNA replication and cell division works the way we think it works, what I just said is true. But can we come up with exceptions? Are there exceptional rare events when we'll get more than one original strand of DNA in the same daughter cell?
The strands can just not separate.
So if the strands didn't separate, what wouldn't happen? Replication wouldn't happen. So you might get a weird cell that didn't undergo division and that still had 2 original copies of a chromosome, 2 original strands. What have we talked about in class that's a process? Yeah, so non-disjunction, trisomy. So if during mitosis there happens to be non-disjunction, you could theorize some sort of a situation where you would have more than one strand of original DNA in a daughter cell. Pretty rare, though, but always good to be thinking about exceptions to rules. Other topics of concern? Yeah.
Could you like go over the nomenclature as to when it's appropriate to use semicolons or slashes and cross?
Nomenclature, yeah. Slashes, semicolons. Which do we use more frequently? In any case where we're talking about a genotype, which of these will we always use? Slash. It's October, it's slasher film month. Look. So what do we use a slash to indicate? The 2 copies of a chromosome. So if you've got that situation, which I know you're already tired of me drawing mom's allele and dad's allele of chromosome 1, if you're diploid, so let's say that's chromosome 1 from mom, we'll use M and D as subscripts in this case. So chromosome 1 from mom, 1 from dad. The genotype would be what allele on both of your chromosomes, both copies of chromosome 1, do you have? So these 2 are interchangeable. The drawing on the left is supposed to be an actual physical representation of the chromosomes. This is the equivalent written out or typed out version. Why do we use semicolons?
To separate it from another gene?
To separate this information from the information we have at another gene in the same individual. So let's say in the same individual, we've got chromosome 2. Chromosome 2, 1 copy that was inherited from mom and 1 from dad. So if we put all of that together into one genotype that represents that individual. The semicolon indicates that we're talking about two separate chromosomes. Two genes on two distinct chromosomes. We'll see later if the two genes we're talking about are on the same chromosome, we don't use a semicolon, but we're not there yet. So slashes separate the different versions of a chromosome. Semicolons separate different genes, different traits. Now it gets tricky. Let's say you've got an individual that's got 4 different versions of chromosome 3. 3A, 3B, 3C, 3D. Now we're not talking about a diploid, now we're talking about a tetraploid organism. How would we write a tetraploid genotype? What would I write here? How many slashes are there going to be? 3. 3A slash 3B slash 3C slash 3D. So you have the number of slashes separates all of the individual chromosomes that you're talking about. You've got an octoploid, 8-copy individual, you're going to have a genotype that has 7 slashes in it, separating all of the different nomenclatures of the original, or the individual chromosomes. Other questions about this nomenclature? I'll be very confused if you give me a genotype and you leave out the slash. And that leads to bad things. So please try to remember every time you write a genotype, put the slash in between the 2 alleles. Yep. Actually, let's go just for a second.
I was going to say, give us an example of a PCR gel.
Can I give you an example of a PCR gel? Yeah. What's your question?
I was hoping you can determining genes inside polar bodies.
Okay, so we've got PCR gels and we've got polar bodies. Okay. PCR gel. Should I draw a hypothetical PCR gel and analyze it? Can I throw a PCR gel on the screen? Let's see. That might take me a second. Let me think about it while we do this question. But yes, we will get to that. So analyzing, interpreting a PCR gel. Okay, so polar body. No, sorry, not polar bodies. What was your question again?
This is that example you did in class of how to determine the DNA inside the oocyte from the polar body.
Right. Good question also. We're going to do that one first, and then we'll get to gel electrophoresis analysis. Okay. So if you're talking about meiosis, and we're specifically talking about oogenesis, production of an egg, what happens after the first cell division? We make 2 cells. One of these cells is going to divide again, produce 2 cells. So here's the oocyte. What's this thing? First polar body or polar body 1. Peanut butter 1. What's the other cell that we haven't labeled yet? Polar body 2. How many copies, let's say we're 2n equals 1, diploid organism that's just got 1 copy of 1 chromosome, chromosome 1. How many molecules of DNA are in the first cell? This is right before meiosis 1, so it's after synthesis.
So the first polar body
we've got after synthesis, so a diploid individual has 2 chromosomes. After synthesis, they've replicated, so you've got 4. Okay, how do those chromosomes divide during the first division? 2 should go into the first polar body and 2 go into the second. And then during the second division, you get haploid gametes. In this case, 1 functional haploid gamete, the oocyte. So we start with 4 chromosomes, sister chromatids in this case, and we end with 3 cells that all together have 4 copies of the chromosome. Physics law about conservation of matter or something, right? Same here. It's conservation of chromosomes. So if I tell you something like you sequenced, let's do it. We'll redraw the exact same figure. So here's polar body 1, polar body 2. Let's say that's the data you get out of extracting the DNA from the 2 polar bodies and counting how many chromosomes there are. There's 1 chromosome in polar body 1 and 2 in polar body 2. How many copies of the chromosome are in the oocyte? Why 1? The law of conservation of chromosomes. Dr. Ross, yeah, new one. Don't make a Wikipedia article or anything about it, though. Right? So 4 total copies. If you know what's in 2 of the cells, you can use subtraction to figure out what's in the 3rd. Is that good, or do you want to go into more detail?
I was like hoping you could the chart.
You want the graph?
Yeah, from the graph.
Okay. So let's say we've got an organism that has 3 different chromosomes. What was on the y-axis? Yeah. Essentially, it's chromosome number. How many copies of the chromosome are in a cell? Is it 0, 1, 2, 3, or 4? Can't have more or less, right? Can't have negative, and you can't have more than 4, hopefully. Okay. So if I drew this, so all of chromosome 1, from its left end to its right end, there are 3 copies of that chromosome. There are 2 copies of chromosome 2. There are 2 copies of all of chromosome 3. Which cell do you think I've sequenced? What's most likely?
First chromosome.
Why?
Mostly just because it has the 2 copies of the 2 and 3, which means that 1 is most likely the one that's
Right.
the non-disjunction.
Right. So most of the chromosomes there are 2 copies of, the majority. And which of the 3 cells, the first polar body, the second polar body, or the oocyte, do we expect to have 2 copies? The first polar body. So what explains the extra copy there? Probably non-disjunction. So now let's say we also looked at the second polar body. Same x-axis, slightly different y-axis. And let's say in blue, we saw what we expected to see. For the second polar body, 1 copy of each chromosome. So is the oocyte that's associated with these 2 polar bodies one that you'd want to use in vitro fertilization? How many copies of chromosome 2? Down here at the bottom, let's summarize what's in the oocyte. Chromosome 1, chromosome 2, chromosome 3. How many copies of chromosome 2 are in the oocyte? 1. Because there was 1 there, 2 there, so there has to be 1 copy in chromosome 2. What about chromosome 3? 2 copies in the first polar body, 1 copy in the second polar body, has to be 1 copy in the oocyte. What about the first chromosome? 0. All of the copies were already identified in the first and second polar bodies. So you might choose to use this oocyte in vitro fertilization, and it's not my job to tell you whether or not you should. I personally wouldn't. I want to optimize the chance that my kids have 2 copies of every chromosome. So we want an oocyte, normally we want an oocyte that's haploid. Here we have no copies of chromosome 1 in the oocyte. Just trying to think if I've got a reasonably fast image of a PCR gel to pull up. I think I better just draw it. So we've got 4 samples. That is 4 DNA samples. We're going to talk about a diploid, as we always do in class, or almost always anyway. Let's say they came from, somebody give me a genotype, single diploid genotype. 1 locus. 2 letters. Big G, little g. Okay. If I knew how to write a little g, I would. Somebody else. Your favorite letter? Too personal? Your best friend's favorite letter. What was that? B. B? Do you want 2 big B's, a big B, little b? Little b, little b? Big B, little b. Okay. Oops. See, I'm screwing up already. Big G over little g crossed to an individual that's got a big B and a little b. Okay. Back to the very first question of the day. This is all the information we've got. What's our assumption about the B locus in the G over G individual? It's wild type. We'll just call it plus over plus for now. And the same is true for the individual at the B's. So it's plus over plus at the G. So we call it G plus over G plus. B plus over B plus. Okay. That's good. Bless you. So let's say we take the B over B, the big B over little b. Now I'm going to use blue. Let's say we self that individual. So we're working with plants. So we have a heterozygote, we self it. Here's the question. Let's say big B produces a large allele, and little b produces a small allele. Physically, talking about gene size, say, number of base pairs in the DNA. What should the results in this PCR gel look like if we take 4 F1 samples and we load them onto that gel? So, well, what's the first thing we should do? If I'm asking you about what do we expect to observe, and it has to do with a cross, you better draw a Punnett square. Okay. So we're going to figure out what do we expect to see if we make this cross? What should we see on this gel? So if we're just talking about a single locus, the B locus, if we have a heterozygote, half of its gametes have big B, half of its gametes have little b. And if we're selfing, that's also true for the sperm, it's also true for the eggs. So you fill it in. So what should the PCR results look like? What would this individual down in the bottom right corner of the gel, or in the Punnett square, look like on a gel? So it would be low, it's small, so it goes farther down from the top to the bottom. So let's say that individual looks like that. Will there be a second band in that well? No, because it's homozygous. So it's only got one size allele, the small over small. I can't tell the difference between the small and the small on a gel because they're the same size. Okay. How many wells have that electrophoresis pattern out of the 4? 1. If we got lucky and we happened to choose the 4 individuals from that generation that represent accurately the expectation from the Punnett square. But that was the question I asked, what do we expect to see? So what does the individual in the upper left of the Punnett square look like? Let's put that individual in that well. Pardon? Closer to the top. How many bands? 1 band because this is a homozygote. So what do the 2 wells in the middle look like? Those are where we load the 3rd and the 4th individuals. There's going to be a large allele, the big B, representing the big B. And there's going to be a small band that's the other copy of the chromosome. And its sibling would look the same. So that's what a gel that represents the expected Mendelian frequencies of those genotypes for that specific cross. Of course, depending on the cross, the gel looks different. All right. Hey, 11:50. So I'm going right over to my office for an hour, for office hours. If anybody has more questions, please come over.